Hi everyone, my name is Dr. Dominique Rodent. I am clinical geneticist specialized in inborn errors of metabolism, and I work in the Department of Human Genetics in Institute of Pathology and Genetics in Charleroi, south of Belgium. I have nothing to disclose regarding this presentation. As an introduction to clinical genetics knowledge you should be familiar with when attending patients with inborn errors of metabolism, we will briefly review what means chromosomes, DNA, and genes. Review the main different types of mutations and their effects on proteins, as well as the patterns of gene inheritance and mutation transmission. And finally, I will discuss genetic counseling for families with a genetic condition and the possibilities offered to these families as prenatal diagnosis and pre-implantation genetic diagnosis. Let's start with chromosomes, DNA, and genes. Cells are the fundamental working units of every living system. The nucleus of cells contains most of the genetic information. The human genome is packed into large units known as chromosomes. Human cells contain two sets of chromosomes, one set inherited from each parent. Each cell, except sperm and eggs, contain 23 pairs of chromosomes, 22 autosomes numbered from 1 through 22, and one pair of sex chromosomes, XX in women or XY in males. Sperms and eggs contain half as much genetic material, that's to say only one copy of each chromosome. Mitochondria have also their own DNA called mitochondrial DNA. Chromosomes are found in the nucleus of all human body cells except for red blood cells. These cells do not contain nucleus and therefore do not contain chromosomes. Chromosomes, in fact, are very long strands of DNA coiled up like a ball of string. Each chromosome contains many genes, the basic functional unit of heredity, and the double helix structure of DNA was described by Watson and Crick, who received the Nobel Prize in Physiology in 1962 for their discovery. The double-strand structure of DNA is composed with a backbone made of sugars and phosphates and a side-by-side arrangement of bases along the DNA strand. DNA from all organisms is made up of the same chemical block units, nucleotides, abbreviated as A for adenine, T for thymine, C for cytosine, and G for guanine, where adenine pairs to thymine and cytosine to guanine, and in RNA, uracil takes the place of thymine. The main characteristics of the nuclear genome is that the double-stranded DNA is a linear structure surrounded by an envelope or membrane. The nuclear genome is very large, as it is made of more than 3 billion of DNA base pairs, but there are only 2 copies of nuclear genome per somatic cells. Nuclear genome has about 25,000 genes, including mitochondrial gene coding for subunits of the mitochondrial respiratory chain. Mitochondrial DNA is much smaller and has mostly coding DNA. It has the particularity to be double-stranded with a light and a heavy strand and be circular. This DNA is not surrounded by a membrane, and is much smaller than nuclear DNA, as it is made up of about 16,000 base pairs. On the other hand, each mitochondria contains dozens of copies of mitochondrial DNA. It's to say that several mitochondria in a cell accounts for thousands of copies of mitochondrial DNA per cell. The mitochondrial genome contains only 37 genes that encode 13 proteins, 22 transfer RNAs, and 2 ribosomal RNAs. Let's go further to explain the different main types of mutations seen in DNA and their effects on protein synthesis. To make a long story short, the process by which DNA is copied to RNA is called transcription, and the process by which RNA is used to produce proteins thanks to a ribosome is called translation. In normal enzymatic reaction, a compound A is transformed into a compound B thanks to the activity of a functional enzyme, which is synthesized thanks to a functional gene. In case of mutation of the gene that encodes that particular enzyme, the enzyme is not synthesized, is truncated, or is not functional, with the consequence of an increase of the compound A upstream of the enzyme deficit and a reduction in compound B downstream of the impaired enzyme. To better understand the different effects of a DNA mutation, we have to remind the way an enzyme is synthesized in normal conditions from DNA and mRNA. As you may know, all proteins are made up of one or more chains of amino acids. One amino acid is produced thanks to the information given by the codon, a specific sequence of 3 consecutive nucleotides on mRNA. A codon specifies a particular amino acid in a protein or starts or stops protein synthesis. In our example, the codon AAG is responsible for the synthesis of one essential amino acid, the lysine. The genetic code is made up of 64 different codons, and more than one codon can specify a single amino acid. Out of these 64 codons, one codon made of the sequence AUG codes for the methionine and initiates traduction of all proteins. 3 codons are the stop codons, UAA, UGA, and UAG codons, and only 61 codons code for the 20 essential amino acids. This phenomenon is called the degeneracy of the genetic code. Amongst the point mutations of DNA, the most frequent mutations are missense mutations. A missense mutation is a change of a nucleotide at the DNA or mRNA level, resulting in a change of amino acid codon, which the ribosome recognizes, hence the production of another amino acid. This amino acid substitution may have no effect, or it may render the protein non-functional. Changes in amino acids can be very important in the function of a protein, but sometimes they make no difference at all, or very little difference. Sometimes missense mutations cause amino acids to be incorporated, which make the protein even more effective in doing its job. But more frequently, it causes the protein to be less effective. In case of a nonsense mutation, the change of nucleotide results in the formation of a premature stop codon. In this example, the A nucleotide is changed into a U for uracil. The UAG stop codon generated by this single base pair change results in the appearance of a premature stop codon instead of a codon specifying an amino acid. The result of this mutation is the production of a shortened and likely non-functional protein. If there is an insertion or deletion of base pair, this causes a shift in the reading frame of subsequent codons. In fact, frame shift mutations have a more dramatic effect on the polypeptide sequence than missense or nonsense mutations. Instead of just changing one amino acid, frame shifts cause a change in reading of subsequent codons and therefore alters the entire amino acid sequence that follows the mutation. That's why insertions and deletions are usually more harmful. In splice site mutations, the normal process of splicing to transfer precursor mRNA into mature mRNA is impaired. A splice site mutation inserts, deletes, or changes a number of nucleotides in the specific site at which splicing takes place during the processing of transformation of precursor mRNA to mature messenger RNA. The deletion of the splicing site results in one or more introns remaining in mature mRNA and may lead to the production of abnormal proteins. When a splice site mutation occurs, the mRNA transcript processes information from these introns that normally should not be included. Introns are supposed to be removed while the exons are expressed. These changes could result in production of a non-functional protein. A last example of mutation concerns large genomic deletions, in which not only a nucleotide but a larger sequence of DNA is left out during DNA replication. Any number of nucleotides can be deleted, from a single base to multiple exons to an entire piece of chromosome. Deletions that do not occur in multiple of 3 bases can cause frame shift. A duplication is the opposite of a deletion. Duplications typically arise from an event termed unequal crossing over or recombination that occurs between misaligned homologous chromosomes during meiosis. The chance of this event happening is a function of the degree of sharing of repetitive elements between 2 chromosomes. The recombination products of such an event are, on one side, a duplication of the site of the exchange, and on the other side, a reciprocal deletion. Population differences in deletions and duplications are also called copy number variations, or CNVs, that are of interest because of their potential effects on gene expression, phenotypes, and disease. Rules for numbering mutations are edited by the Human Genome Variation Society. In this example, at the level of the cDNA, the mutated nucleotide is on position 10, where a G is replaced by an E with a consequence of a change of amino acid. At the level of the protein, the annotated glycine is replaced by a serine on position 4. Now we have understood mechanisms of some mutations and their effects on proteins or enzymes. It's time to understand the patterns of gene inheritance to appreciate how mutations or genetic conditions are passed on through generations. Diseases caused by mutation in a single gene are usually inherited in a simple pattern, depending on the location of the gene and whether 1 or 2 normal copies of the gene are needed. This is often referred as Mendelian inheritance because Gregor Mendel first observed these patterns in garden pea plants. Single gene disorders caused by DNA changes in one particular gene often have predictable inheritance patterns. If the mutation affects one of the 22 autosomes, the heredity is said to be autosomal dominant or recessive. In case of inheritance linked to X chromosome, this could be recessive or dominant. And in case of mitochondrial inheritance, the transmission is from the mothers. One has to keep in mind that most inborn errors of metabolism have an autosomal recessive pattern of inheritance. In case of autosomal recessive inheritance, both parents are a carrier of a mutation or a trait, represented by the red box on this slide. For this couple of carriers, at conception, each SIP has a 25% risk of being affected, a 50% chance of being just a carrier as their parents, and a 25% chance of being unaffected. When we draw the pedigree of a family with an autosomal recessive disease, as this in in this example, females are represented by a circle and males by a square, we observe that only the patient who inherits both mutated traits, represented by a big A on this slide, have the disease. And this patient is said to be homozygote. Carrier patients' parents are not affected and do not have symptoms. Of course, the couple of carrier parents has a 25 risk of having another affected child who will inherit two mutated traits. In case of autosomal recessive inheritance, in general, we observe other affected members in collaterals of the pedigree, but not on several generations as seen in autosomal dominant inheritance. Unless the affected patient marry a cousin or someone from his family, the risk to have a child affected with the same condition is almost non-existent. Transmission is not related to sex of patients because mutations are carried by autosomes and not by sexual chromosomes. Offsprings of consanguineous unions may be at increased risk to genetic disorders because of the expression of autosomal recessive gene mutations inherited from a common ancestor. The closer the biological relationship between parents, the greater is the probability that their children inherit identical copies of one or more detrimental recessive genes. The risk is higher than in general population and reach 6.25%. Of course, in case of consanguinity, first consider autosomal recessive disorder, but always keep in mind that other patterns can also occur in the family tree. In case of autosomal dominant inheritance, dominant mutations are expressed when only one copy of that mutation is present. Therefore, anyone who inherits one dominant trait, such as a mutation for acute intermittent porphyria, will have that disease. The risk of transmission of a dominant trait to next generation is 50% and is not dependent on the sex of the child. If you analyze the pedigree of a family with a dominant trait, you often highlight that an affected patient has at least one affected parent. However, dominant mutation can also happen in an individual for the first time with no family history of the condition. We call them a neomutation or spontaneous mutation. Dominantly inherited genetic diseases tend to occur in every generation of the family, with a transmission risk to offspring of 50%. Dominant traits affect males and females equally. Diseases caused by mutated genes located on the X chromosome can be inherited in either a dominant or recessive manner. Since males only have one X chromosome, any mutated gene on the X chromosome, dominant or recessive, will result in a disease. Because females have two copies of X-linked genes, they will not be affected by inheriting of a single recessive mutation on an X-linked gene. For X-linked recessive diseases to occur in females, both copies of the gene must be mutated. Families with an X-linked recessive disorder often have affected males, but rarely affected females on each generation. If the mother is carrier of a mutated trait, the risk to transmit this mutation to her sons is 50%, depending on which X chromosome she transmits to her baby boy. If she transmits the mutated trait to her daughter, the baby girl will be a carrier. It's to say, an unaffected child on the clinical point of view. On the other hand, if the father is affected, he will never pass or transmit his mutated X to his sons. It means no son will be affected. The affected father will only give birth to a carrier baby girl or daughter. If we take the example of a family where two male patients with Hunter disease are diagnosed in the family, in black on the slide, these two patients are linked by a carrier and asymptomatic woman. In this family, it is important to identify carrier women to give them a correct genetic counseling the day they want to be pregnant. A carrier mother has a 50% risk to give birth to carrier daughters and a 50% risk to give birth to affected son, depending on which chromosome she transmitted to her son. Every baby girl from an affected father of a X-linked recessive disorder will be a carrier, and every son of an affected father won't be affected as he receives his X chromosome from a healthy mother and a Y chromosome from his father. However, for X-linked dominant diseases, a mutation in one copy of an X-linked gene will result in disease for both males and females. Families with an X-linked dominant disorder often have both affected males and affected females on each generation. A striking characteristic of X-linked inheritance is that fathers cannot pass X-linked traits to their sons. Fathers only pass X chromosomes to their daughters and Y chromosomes to their sons. In contrast, affected mothers pass X-linked genes to both sons and daughters equally. As an example, let's take the pedigree of a family with Fabry disease. On the first generation, the affected male has no affected son because his son received the X chromosome in blue from his healthy mother and a Y chromosome from his father. Every girl of this affected male also got the mutated X from their father and are then also affected. Because in Fabry disease, females are usually later and less symptomatic, but they are no more simple carrier. They are symptomatic sometimes with symptoms as severe as symptoms seen in males. On the third generation, 50% of the daughters from an affected mother receive the mutated X and 50% of the sons are affected. Carrier females are no more carrier but are symptomatic and affected, as said before. Females and males are affected equally in the pedigree, and there are affected patients on every generation of the pedigree as in a dominant disease. Last one, but not least pattern of inheritance concerns mitochondrial inheritance. In this case, we speak of a maternal transmission. It's to say that every baby of a mother inherits mutated mitochondria and its mitochondrial DNA from her or his mother. And this transmission is not related to gender of offsprings, because both females and males receive mitochondrial DNA from their mother. Symptoms can appear on every generation in a family, but always connected to a mother transmission, because a male never transmits its mitochondria to his children. Why is it like that? While male sperm contain 100,000 paternal mitochondrial DNA, these do not enter the oocyte, degrade in the oocyte, or are significantly diluted among the abundant maternal mitochondrial DNA in the oocyte during fertilization. On the other side, the oocyte contain mitochondria with 100,000 of more copies of maternal mitochondrial DNA. And in the total, the fertilized zygote contain nuclear DNA derived from both parents, but all or essentially all mitochondria containing maternal mitochondrial DNA are inherited from the mother. Which are the objectives or goals of genetic counseling? If we go back to the definition given by Harper some years ago, genetic counseling is the process of providing the necessary individualized information about a genetic condition and its implications, and to assist in decision making in a non-directive manner. With this definition, we understand that the ideal genetic counseling should be composed of, first, a trained professional who understands genetics and its ethical implication. The information provided to the patient and his family should be relevant and objective. It's to say, sometimes with documentations or literature references. And also the counselor has to be sure that the counselee or the patient and his family perfectly understood the information provided. On this point, psychologist support is very important because the patient has to debrief with the psychologist what he understood under the emotional situation of the announcement of a diagnosis and the risks for his family. So before prescribing a genetic test, the patient has to sign an informed consent, and of course the genetic information provided during the consultation stay between the counselor and the counselee. It means that confidentiality is very important in terms of genetic counseling. So the geneticist draw a family pedigree with the patient and consider which are the risks for the patient itself or for his parents or his children, his aunt and uncles, etc. What is important is that the counselor handle on an appropriate way the potential risk of discrimination of the testing. So altogether, well, the patient has to be autonomous when he takes a decision for himself, and this decision shouldn't be influenced by beliefs or thinking of the counselor. Which are the indications for a genetic consultation? Sometimes the couple comes to genetic counseling before pregnancy in the frame of preconceptional genetic counseling to analyze the factors or genetic diseases that run in the family, for example, in one of the partner, that might affect baby during infancy or childhood, because of history of infertility, multiple miscarriages or stillbirth, because of previous pregnancy or child affected with a birth defect or a genetic disease, and also to be informed about assisted reproductive options now available for them. The couple can also come to genetic counseling during pregnancy because of an abnormal test made in the mother, for example, a NIP test, because of an abnormal ultrasound, for example, for a cardiac malformation, or because of amniocentesis has to be done because of the risk of recurrence of, for example, a recessive disease in the family. The parents can also come to genetic counseling after birth because of an abnormal newborn screening test, because of birth defects in the child, because of developmental delay or intellectual disability, or even autism, and also suspicion of inborn errors of metabolism. Prenatal diagnosis is discussed within the frame of the genetic counseling consultation. First, to give the most complete information to couples' parents about the genetic disease present in the family and the risk to have an affected child, considering the family history, the pattern of inheritance of the disease, and or the detection of prenatal malformations. Also to permit an early detection of a genetic condition and take decisions in a non-directive manner about prognosis of the pregnancy, possible treatment options, and reproductive assistance options too. It's important to discuss about alternative arrangements as adoption, surrogate pregnancy, gene therapy, and so on. An accurate diagnosis is important for medical management, determining prognosis and genetic counseling for future pregnancies and CIPSHIP. Classically, as methods for prenatal diagnosis, we have non-invasive methods as antenatal ultrasounds. We have also invasive procedures as fetal cord blood, chorionic villus samplings, or amniocentesis. In case of inborn errors of metabolism, most prenatal imaging findings are non-specific and often observed in the third trimester of pregnancy. An inborn error of metabolism is suggested by the association, for example, of acid and hydrops fetalis in lysosomal storage disease. In case of intrauterine growth restriction, also disease of metabolism could be suggested, as well as central nervous system abnormalities as anagenesis of the corpus callosum in PDH deficiency. Echogenic kidneys as well as epiphyseal striping could suggest a paroxysmal disease, and craniosynostosis and a wide spectrum of dysostosis also suggest an inborn error of metabolism. On the right-hand side of the slide, a picture of an in utero hypertrophic cardiomyopathy, which was confirmed to be an infantile POM disease after the amniocentesis at 22 weeks of pregnancy confirmed the presence of a mutation in the gene of maltase acid. An invasive procedure for prenatal diagnosis concerned the CVS or chorionic villus sampling, performed usually between 9 and 11 weeks of pregnancy. And the methodology to make this kind of prenatal diagnosis is to make a punction of chorionic villus through abdominal or vaginal route under ultrasound guidance. Of course, the risk of miscarriage of this procedure is ranged from 0.5 to 1%, but this technique permits an early diagnosis and early decision concerning the prognosis of the pregnancy. Amniocentesis is performed a bit later in pregnancy at around 15-16 weeks of gestation under ultrasound guidance with a risk of miscarriage a bit lower. The aim of the technique is to punction amniotic fluid through the abdominal wall to have a minimum of 15-20 milliliter and to centrifuge the liquid to separate fluid and cells. The supernatant is then frozen for further analysis, and fibroblasts from the fetus are put in culture for further analysis, as well as biochemical or DNA studies. The supernatant of amniotic fluid is useful for the analysis of metabolite profiles. For example, glycosaminoglycans, oligosaccharides, sialic acid, lysosphingolipids, and some enzyme activities can be measured for lysosomal storage diseases. As well, 7 or 8 dihydrocholesterol, desmosterol, and latosterol can be useful for detection of cholesterol synthesis defects. Acylcarnitine can be measured by MSMS in case of fatty acid oxidation suspicion, as well as organic acid for organic aciduria and polyol for transamylase deficiency. Measurement of enzyme activities for most inborn error of metabolism can be done on cultured amniocytes as well as whole cell assays or metabolite measurements. Molecular karyotype by array CGH or shallow sequencing, as well as gene, exome, or genome sequencing done with the next generation sequencing technique can be performed after extraction of DNA. Amino acids can be measured after fetal cord blood is done, for example, in case of suspicion of serine deficiency. For couples with a history of genetic condition, pre-implantation genetic diagnosis is a reproductive option. Pre-implantation genetic diagnosis has been introduced as an alternative to prenatal diagnosis. With this technique, the genetic diagnosis is made on an embryo obtained after in vitro fertilization. The aim is to transfer an unaffected embryo in the maternal uterus. This is a kind of early form of prenatal diagnosis offered to couples at risk for transmission of a genetic condition within the frame of genetic counseling. And the aim is to prevent the genetic transmission of a severe genetic disease. Of course, cultural beliefs and religious practices have to be taken into account regarding the acceptance of this method of reproductive assistance. Intracytoplasmic sperm injection, or ICSI, is a revolutionary assisted reproductive technique used in conjunction with in vitro fertilization to achieve fertilization. With this technique, a single sperm is injected directly into the cytoplasm of a mature oocyte. After ICSI and in vitro fertilization, the embryo is usually biopsied at the stage of blastomere, it's to say a 8-cell stage, generally obtained at day 3. At this stage, one cell is withdrawn for DNA extraction and further genetic analysis to check if the embryo is affected or not and if the embryo could be reimplanted in the maternal uterus. PGD is a long procedure. That's why the success rate is estimated to be between 20–25% and is limited to strict indications where the mutation is known in the family. In our example, the 10 oocytes at pickup on D0, for example at D1, there are 2 lost eggs after fertilization, and on day 3, 6 embryos are available for biopsy. After extraction of one cell and DNA analysis for genetic diagnosis on day 4, in our diagram you see that one embryo has no result, one is affected and can't be transferred to uterus, one is unaffected but has a bad morphology and either can't be transferred. So in total only 2 embryos are unaffected and can be transferred in maternal uterus on day 5. So the pregnancy success rate of this procedure is estimated to be 20–25%. In X-linked diseases, sometimes the only way to avoid transmission of a devastating disease is the sex determination on embryo to select which embryo should be transferred to maternal uterus to have a healthy offspring. For example, in case of X-linked adrenoleukodystrophy, if the father is affected, the only way to have a healthy baby is to have a baby boy, because as you know, the boy inherits the Y chromosome from his father and not the mutated X, because the X is inherited from his mother. As summary, genetic counseling should be part of the management of every patient with inborn error of metabolism. During the diagnostic phase, the geneticist helps to orientate the genetic tests to perform and their interpretations, depending on the severity of the mutation and their interpretation. Genetic counseling assists individuals in understanding test results, but also their consequences for themselves and the family. The genetic counseling therefore deals not only with the medical aspects of a genetic disease, but also with its social and psychological consequences and possibilities and strategies for coping with genetic diseases and genetic risks. So ethical, social and cultural issues related to clinical genetic testing and counseling have to be taken into account during the whole process, as cultural beliefs and religious practices influence uptake of information and understanding of genetic disease.